Let us now begin the new section with Alejandra Ferenzi. She's an agronomist, has a master's degree awarded by the University of Michigan. She's a biosafety manager at the Ministry of Cattle and Fisheries. Good afternoon or good morning, everyone. Today we will be talking about genetically modified organisms and how they are regulated in Uruguay. What is an GMO? It's a live being that has genetic material that wasn't part of originally of its genome, and it includes different technologies. We will not go into detail, but they include recombinant DNA. And how would we create a GMO? First of all, we need to create the desirable characteristic. These techniques are beneficial because we can find this feature in any organism. For instance, if you want to modify a vegetable, we can find the feature in any other organism because the genetic code can be read by any organism. So the first step is to identify that desirable trait. Then we should locate the genes in the source organism. We need to isolate them, clone them, and to conduct the necessary reconstruction in order to insert them into our target organism. We have recombinant DNA, and through transformation, we introduce this genetic construction into vegetable cells. After this, we will select in which cells the new construction has been inserted. Then the cells are regenerated and create a new whole plant. We need to consider the totipotency capacity of vegetables, and we can actually regenerate the whole plant. And then we need to have an improvement program in order to insert these genes into the elite varieties from an agricultural perspective. The aim of this talk is to understand how we have created an GMO or transgenic organism and how this is regulated and how these developments are regulated in Uruguay. This is why we need to also consider the regulatory system, which needs to be done before selling them. This is actually, we have a huge jigsaw puzzle and a lot of teaching material as well online where you can find this process in further detail. For instance, different transformation, genes. These are all process details, and if you want to study the matter further, you can find more video clips and different types of material on the creation of a GMO. I also, please remember that transgenesis has other previous stages as well. For instance, random mutagenesis in the genome in order to reach the desired trait through radiation, insertion, or chemical agents. These mutations from these methodologies, radiation, insertion, or chemical agents, happen randomly. These mutations just occur, and then we analyze the genotype. Also regarding transgenesis, we insert that specific construction, it happens randomly. We do it randomly in the genome, and this is why we have this guy who is blindfolded. This justifies the need for a regulatory system because we need to see where this gene is now located. It can't interrupt other constructions or other gene expressions. Nowadays, there are products, new biotechnological products. Have a look at this in orange. Genome editing, target and mutagenesis. They allow us to make a specific change in the genome. There can be specific mutations, replacing a sequence with another one, insert a new sequence, etc. We can edit the genome and we can work with the genome more accurately. This is something I wanted to tell you about because it is worldwide. There are now products in this regard, and the various countries are working on how to regulate this. The regulation for genome editing is not ready in Uruguay yet. We're working on it. Now we will explain how in Uruguay we regulate GMOs. These are organisms created by implementing the different steps I was telling you about, transgenesis or recombinant DNA. Regulations. We would like to give you some more information about Uruguay in the world compared to the countries that have adopted this technology, having genetically modified vegetables. Here you have 29 countries that sow GMOs along 190 million hectares, which is the area in green on the map. Have a look at Uruguay is in the 11th place in the list because we plant at least 1 million hectares. And at the bottom, there are other countries where they plant less than 1 million hectares. It's also important to remember that if we, in the world, we add the 29 countries and the countries that import biotechnology-derived products for human and animal consumption, we would be adding 43 countries. So in total, we should have 72 countries that have a regulatory system that authorize accelerated human or animal consumption of these organisms, or the growing as well. In light green, you can see the countries that import or approve these events, and in dark green, the countries that grow these crops. This is the same list with the main countries. Uruguay is highlighted in red. Additional information. What type of species are authorized in Uruguay and in every country in the list? In Uruguay, maize and soybeans are authorized commercially for growing and selling, and there are other crops in other countries that are approved as well. On the left, you have a link to the website where we post all the events approved in Uruguay. Also the feature provided to the species and the events that are being analyzed. So in which stage of the analysis we're at. Regarding Uruguay, we have requested applications for use for vegetables, and we also regulate this use at a greenhouse, at nursery level, and this is actually confined a confined program. We have a specific protocol for the labs and nurseries. Then we have field tests, and then we have seed production with a safety protocol for exporting purposes, and that includes over 1,000 hectares. And then we have the commercial use category, where events are not regulated. As I was saying, commercially, Uruguay has maize and soybean events. We have around 12 events authorized for soybean and 20 for maize. Characteristics. resistance to insects, and tolerance as well. And these are the different combinations, tolerance to different herbicides and insects. How are GMOs regulated? Uruguay has had a regulation for 24 years now. Of course, we produce agricultural goods, so already from 1996, since 1996, we have been working on this with the first soybean that was resistant to herbicides. And the idea was to define if its use was authorized or not. And Uruguay works towards this regulatory system in order to accompany technological changes. Currently, we have the 2008 decree that is part of biosafety system that aims to work with the management and use of genetic GMOs so that they are safe for animals and humans. Why biosafety? Within biosafety, we can include all the political measures and procedures that aim to preserve the environment and to protect the health of humans and animals in different situations. Food production, introduction of pests and animal diseases, etc. Biosafety will depend on each country. And sometimes it includes all the different aspects, but here it refers to different areas. I work with the biosecurity of GMOs. How does the system, the national biosecurity system, determine the authorization of a transgenic event in Uruguay? We apply the risk analysis methodology, which is internationally accepted to analyze any kind of new technology. This technology is well established, and at the Codex Alimentarium, we actually we base our work on different international agreements, including the Codex. As you can see, this methodology includes 3 steps or areas. For instance, someone cannot work in 2 areas, but of course the work is coordinated. We have management, evaluation, and risk communication. The decree we have is easy to explain. It helps us explain risk analysis because it places all the participants within this technology. Please have a look at these 3 circles organized according to how Uruguay regulates GMOs. In blue, you can find all of us in charge of the technical analysis or risk evaluation. We analyze the applications, then we have 9 participating institutions and their experts. We work with environmental issues and potential impacts on natural enemies. Also, for instance, human and animal health, non-target organisms, etc. It depends on the group that analyzes the application. Then we write a report that includes all the technical and scientific information, and then we go on to the managers section. The ministries are here are the top authorities. In Uruguay, there is a national biosecurity cabinet formed by 6 ministers. The president is the Minister of Culture, but we also have industry, economics, foreign affairs, health, transport, and agriculture. There is a commission for risk management. They lead the process. and they write a recommendation report to the ministers by including the contributions of the technical step. There is also public consultation because the reports are disseminated and they're communicated to the society for a month. And there is also a social and economic report prepared by the Ministry of Agriculture that includes relevant issues. The Commission takes all these elements and writes up a recommendation report for the Cabinet, and the Cabinet takes the final decision. Remember, I was showing you the website where we approve the events. There you can see in each instance which are our competences and how we organize our work. Okay, if I know that there is an event, how do we address it? We characterize risk and we need to distinguish it from danger or hazard, because a danger has a potential to cause harm, a shark in the water. But if the person doesn't go into the water, they will not be exposed to the danger. So we define what we want to protect. We list the potential dangers or hazards. We analyze the exposure probability, and we evaluate the risk, this probability of this danger actually causing harm. Because there is no zero risk situation, but we can classify different risk levels. The danger can be present, but in Uruguay we can mitigate this danger. And we can handle this danger so that it doesn't create a risk. Let us now go back to the first image. An application is sent and risk is assessed. The experts write up a list. What can go wrong? Which is a list of dangers. What can happen if we were to release or consume this type of GMO? So we make a list and then we analyze a risk hypothesis. And we analyze the probability of that danger actually taking place and doing harm, and also its consequences. This is a generic table. We conduct a qualitative analysis based on these charts, where we have the harm, the probability of doing harm and its consequences. Based on that, we describe the risk and we provide this information to the managers who will decide where the line is drawn. As I was saying, the risk, 0% risk does not exist. Accidents can happen all the time, but we need to think about what the probability of something happening is and its consequences as well. So managers tell us, is the analysis sufficient or not? They might ask us to analyze more information. And this is where we also work with managers, which is a technical and political aspect. They need to tell us what they want to protect, which the terms of reference we have for the analysis, and then they add other social, legal, political, economic aspects for decision making. For instance, a very important aspect for this methodology to be successful is risk communication. This is where we exchange opinions, information, and of course people need to know about this, but we also need to guarantee the transparency of the process. This might take 2 or 3 years, even more in some cases. So the communication needs to be recorded, especially what happens in each stage. And when the final decision is made, there shouldn't be any kind of surprises for other stakeholders. The necessary guarantees need to be in place. Of course, we are open to any questions you may have. Before we finish, I would like to thank the INASE for the help with the biosecurity system, because they participate with specific experts at the ad hoc groups. And also regarding the protection of biosecurity in Uruguay and controlling the protocols, which happens when they authorize research tests, or when when seeds are produced in large areas. For both situations, there are specific protocols. These are the so the INASE's biosecurity area is in charge of that, and we are in close contact. Also, at molecular laboratory level, we analyze seeds and we try to detect transgenic events that need to be controlled because they are not authorized in the country, or they should not enter the country through seeds. So that's what I had to tell you about, and I remain at your disposal in case you have any questions. Thank you. Thank you, Alejandra. Your presentation was very clear. It is clear internationally that GMOs are more affected throughout the world. And actually, this is the case of our system, because we can help provide the necessary guarantees. We have some questions on the chat from Iraq. Which is Uruguay's criterion to reject or accept new GMO applications? What we do is, actually it depends on the use, but we analyze the applications environmentally. We request information about environmental assays and also molecular characterization. On the website, you can see the form. We can translate it as well if you need, if you need it in English. There you have our molecular requirements. For instance, just a single gene copy. It shouldn't be for instance, you need to include which kind of species is being affected, that they shouldn't affect our own native crop, for instance. If the feature is insect resistance, maybe we see it shouldn't kill other beneficial species. So it's a long list, but the idea is to is that the decision is science-based. First of all, if there is no significant risk for health or for the environment, then it does go on to the next stages. If a problem is found and the process is halted until the problem is solved. Okay, that's very clear, and here you have the information as well. Another question, is there harmonization regarding the region's application processes? We have been trying to do this for a long time with the region. The idea is to harmonize the technical criteria that we use to analyze the applications. And then in every country, there are different managers that take into consideration different aspects, which depend on each country.
So we should reach a harmonization. And if you see the lists, we try to reach that harmonization. We try to have more or less all countries, but for instance, Argentina, Brazil, there are 40 events authorized in corn, in Paraguay 25, and Uruguay 20. So we have a commission within the Mercosur to analyzing these differences, and we are realizing that many times, even if 40 to 20 is a difference, but those 20, not all of them happened in Uruguay. So they said, I want to grow, to develop this crop, and so there we realize about differences that were not so important. So very clear your answer. Thank you very much, Alejandra. And now we are going to invite Mariana Menoni. She is a bachelor in biochemistry, and she is studying her PhD in the University of the Republic in Uruguay. She is responsible for the molecular health sector of the seed quality lab of INASE. She's going to tell you about the work we are using. So thank you very much, Vanessa, for your presentation. The title of my talk is the set of molecular tools you always wanted to have. And what I'm going to do is to tell you what are those molecular tools that we have in the INASE lab. And I'm going to talk a little bit about each of them. So to start, and make a brief introduction and level some basic concepts, I wanted to tell you, show you this scheme that I am explaining with the drawing of a plant that if we look with the microscope, we can see it has a vegetable tissue. And then if you go deeper in the cells, we can see there is a nucleus. Inside the nucleus, there is a DNA in the form of the chromosome. The DNA has some bases with nitrogen, this guanine, adenine, cytosine, thymine, and so on. And then that's how we read the DNA sequencing. This DNA, if we think about a living cell, it has the information, the inherited information from one generation to the other one. And the unit of this information is saved in the gene format, to say it some way. And this is translated into protein. That's what we can observe, the molecule that can express what you have in the DNA. With the example of Alejandra with GMOs, if we have a GMO with some special modification in the plant, it will bring another feature that gives an advantage in comparison to the crop that doesn't have that genetic modification. After this basic introduction, I wanted to tell you that the main tools that we use in the lab routine, they are based in those that let us detect modifications in the DNA. And then the best technique is PCR or polymerase chain reaction. And then we have other tools that let us detect this protein that was coded in the DNA. And then we have ELISA technique that is representing enzyme-linked immunosorbent. And then the reactive, the immunostrips that are helping to diagnose kind of quickly and immediately. So having explained both techniques, so I would like to tell you briefly how they work and how do we use them at the lab, at the INASE lab. So to start, I would like to show you the immunostrips that we use to detect GMO proteins in crops. And they can be used starting from seeds or from leaves, vegetable leaves. And we have to make the tissue uniform, and we mix it with water, and there we take the sample, and there we put the immunostrips, and there if we have two lines, it's positive. If it has one line, it's negative. And this shows that it is working. It's an internal control with these immunostrips. This tool let us have a quick diagnosis, and it doesn't need require any lab equipment. It can be done in the field because it's enough to have a tube to prepare the mix and just use these strips. Certainly, we have to buy the proper immunostrip to detect the protein we are looking for, and then we can detect any GMO that is expressing that protein. So moving to the techniques that require a minimum equipment, lab equipment, we have the ELISA and it's based on the antigenous antibodies technique. So it's we put on a plastic plate the antibodies that we want to detect, and they are incubated, and this is revealed with these antibodies that are specific, and we see which are the positive samples. So with this way we can detect proteins expressed by GMOs, and we use it in the lab, an analysis that we call trait purity, and we detect the percentage of seeds that are expressing that feature or the characteristics. We do it starting from an individual seed. So we put one seed in each of the spots of this plaque, and that's how we estimate the percentage in a semi-quantitative way. Another application of ELISA is to detect viral proteins that let us detect some vegetative propagation material that happen in the potatoes, in the grapes, and also in fruits that are not perennials. And we need, in this case, to have specific antibodies for each of the viruses that are identified in each species. Up to here, we had the techniques to detect the expressed proteins, and then we have tools to see changes in the DNA level. So in this case, PCR is the top star to analyze. It has been it has been the trend because of the COVID-19. So it lets us find out about a DNA specific event, and it let us find the primers. And according to the reactives or the primers that we use, we can identify and the results that we can find. So we have detection of GMOs by PCR, and depending on the primers that we use, we can develop different strategies. For instance, we can detect a specific transgenic event, and for that we need the specific primers. And we have in INASE all these specific transgenic primers for all the ones approved in the country, and also the ones that have partial approvals or with biosafety measures. We can also detect a gene that is providing special characteristics, like the gene EPSPS that tolerates glyphosate. And then we can, it's important to monitor colza and alfalfa imports, where we don't want them to come with elements that are tolerating glyphosate. So we can detect this these characteristics, and this is saving us many reactions and many studies that we should need if we didn't use this. Another approach that we can make is to detect GMO contaminations in conventional lots. And many times what is used are screening methods that are most of them in the GMOs, like the promoter 35 and so on, that are common to many events. And then we can detect the presence of the P35S promoter, sorry. So we need a more advanced lab for this type of studies. We have to take a DNA sample, and we need some special equipment and tools, and also training for the analysts to be able to do it. Another application of this PCR is the detection of the bacteria Xanthomonas citri that is creating the citrus canker, sorry, that leads to reduced production and reduce the value of the fruit that is produced. So from INASE, to support the certification scheme, we try to certify from the leaves to try to find this pathogen before gets strong and it's spreading. And then we keep the gene bank free of this pathogen before they come to the field and it becomes a real important health and economic problem for the sector. We also, in the lab, we have deployed the identification of the weed Amaranthus palmeri by PCR. This is something we have been doing in some study that I was carrying out for the end of the career. It's important that this weed is reproducing very quickly, and once it's installed, it's difficult to handle it, and it leads to big losses, productive losses, because of the damage that it causes. And the main way of spreading of this weed is through the seed. So we have to find it in the seed lots that are traded before they go and are sown. And the difficulty of this weed is that it is very difficult to identify it, because there are other species that are related to it and they have the same morphology, and it makes it more difficult to identify it. So that's why to have this analysis technique by PCR, that gives us some, let us know that if we identify something in a lot, that it could be this. When we do the PCR, we can be sure, and then we can discard the lot, or at least avoid that is coming to the field. Another option of PCR is the PCR in real time. That involves another equipment, a more expensive equipment, and it's based on the same principles as a PCR. So in this case, it is an equipment that detects the inflorescence and let us identify the DNA specific, and we use it to identify GMOs to see what's the percentage of GMOs present in a seed lot. What we need to know is what's the transgenic event that we are looking for. We need the reference material that is to create a graph or a chart, and then we can make it this calibration graph, and then after the analysis we can find out which is the number of copies of DNA in this GMO, and establish the percentage of GMOs. This is the importance of having a good sampling. I'm talking about a sample we are quantifying of the DNA. We are transferring that to the seed lot, and it has to be, the sample should be representative of the lot. In general, this technique is used to detect low amounts of GMOs, and we have the needed reactives to identify all the GMOs that are there. So, and finally, but not least important, the identification of varieties with the SNP markers. A SNP marker is a specific change in the DNA. For instance, if we take a look at this diagram, in the upper part of the slide, we have the DNA sequence of 4 varieties, A, B, C, D. And if we read it from left to right, we see it's identical for all of them, except for the SNP1, where A and B, they have a T, and B and C have an A, an adenine. So if we continue reading the sequence, in the SNP2, we have another difference. When A has a C and the other 3 have a T, and so on in the rest of the SNPs that we find along the sequence. And so that way, what we can do is generate an ID profile for each variety, as you can see in the chart on the right to the bottom, to see which is what it is having in each of the SNPs, and then have like a fingerprint of each of the varieties. What we did in the lab, and thanks to the support of the private sector, is build this matrix for soybean, all the soybean that is traded in the country and has been registered. And this way we have the specific reactors using PCR in real-time PCR. And that's how we can identify the varieties for soybean using these SNP techniques. And finally, to finish, I wanted to present a summary of the different tools that we use in the lab that let us assure quality, genetic quality, health quality, and gives warranties to the country that the seeds that are traded, they are high quality seeds. And in this case, I say one more time that we count with ELISA technique that detect protein, and then we can determine trait purity. We can detect viral proteins, and then that way assure this health quality, the immunostrips. Then using PCR, we can detect GMOs, quantify them in real time, and also detect pathogens like the one producing citrus canker or the pulmonary weed, and then also the varietal identifications through SNP. So this way, I put all the sets of tools that we have. Sorry, I forgot to tell that for this, we are using all the protocols that are validated, that are and published by reference labs from the EU. Everything is standardized and it's not random. So we have controls of all those techniques. We have positive and negative controls to be sure about the results that we are reaching. And so here I'm come to the end of my presentation, and really you have a set of powerful tools, and this let us so it's good that you can keep this presentation so that you get to know what you are able to see all we do at the INASE. So there is a question to complement this this presentation, and taking into account other crops that can be identified with DNA. Is INASE thinking about deploying this to do other things of production? We are using it for soybean to control, and we are open to use it for other crops. We are not doing it yet, but yes, we are open to see what's the demand of the sector and how we could make progress in this regard.
What do we need to set up a molecular biology lab? Okay, we would need a whole talk actually to talk about this. But basically, I would say that we need a PCR method in final time or real time, depending on the type of analysis you would want to conduct, and also to have a specific lab infrastructure, which is very important in order to prevent cross-contamination of samples, and to have an amplification area, a DNA area. And of course, we should buy the reactives and basic lab equipment. And we should also be in touch with some analysts and other analysts. Thank you so very much, Mariana. Now I would like to thank you to the final presentation of this section. We welcome Rosina Fossati, who has a BA in biology, awarded by the University of the Republic. She studied at the Davis University on animal science. She is the co-founder of Genexa, evolutive DNA, and she is now the company's CEO. Thank you very much, Rosina, for being here with us today. I would like to give you the floor. Thank you, Vanessa, for organizing this meeting. Thank you, Inasi, for this event and for the invitation. It's an honor for me to be here today. I would like to make a presentation on genetic fingerprinting and what we mean when we use this term. As Mariana has already said, DNA contains all the genetic material of all life beings, bacteria, viruses, people, everything is included in the DNA, which is included in the nucleus of each cell. It has a double thread with the cytosine, guanine, and all the other elements. It is the combination of these bases creates unique individuals. DNA is represented by conserved regions, even among individuals of different species. And there are other variable regions that are the most powerful ones for us, and we will be focusing on them for individual identification purposes. So what do we talk about when we talk about genetic fingerprinting? Many times people come to the lab and ask us about, I don't know, having a DNA test on an animal, on a piece of clothing, etc. And people don't know what they mean when they say DNA. It's the analysis of several regions in the DNA that actually create a unique biological identification number. It's a sort of genome barcode, and it is inalterable throughout life. If we go to the supermarket and we want to buy skimmed milk, and there are two brands, each type of milk will have a unique barcode. And when we pay for the milk, we won't read the whole list of ingredients. We won't have a look at the box, etc. We will just have a look at the identifying barcode. Something similar happens to DNA. We pay attention to specific regions, which are very powerful for identification purposes. Molecular markers used for identification, besides SNPs, it's the ones that are called SSRs. This type of marker is really valuable because it is extremely valuable. It varies from person to person, and this variation lies in the number of GT repeated runs. For instance, the DNA molecule is set in a specific way, and then GT is repeated 6 times. This is a type of nucleotide satellite marker. There are other trinucleotide markers. They can have 4 bases. These are actually repeated in tandem. In green, you can see a marker, a bovine marker, BM2113, where we can see just 1 peak. So the repetition, 135 pairs of bases are repeated. In this case, it's a homozygotic animal, therefore it received one of the alleles from the mother and one from the father. On the right, there is a barley marker, where you can see 2 types of variants, 98 and 102. This means that the plant received one of the paternal alleles, any of them, and the other one was received from the mother plant. What is important in this example is to see the gray bars, which are the other possible barriers, markers here. And these microsatellites need to be variable, and they need to be able to distinguish between one organism and the other. In the case of bovines, and animals can have, there's a high chance of one animal having the same allele in one marker. Therefore, we need to analyze a marker panel in order to accurately identify an animal within a sample. It is usually 10 to 15 samples are analyzed. Markers are analyzed per sample. These are inherited from parents to children. They are easy to measure. They depend on the person. This depends on the operator's experience as well. They're not as easy as the SNPs because we have two possible results. Microsatellites are not so nice. It depends on the DNA quality and quantity, if it is degraded or not. It depends on the operator's experience and expertise as well. Once we have a solid and validated system that is reliable, then through our current platforms, we can include several microsatellite markers within one tube. In the animal sector, there is an international animal genetics organization that agrees on the validated panels and works with laboratories as well, so we can have an agreed on panel for each species. And that is essential in order to compare DNA that comes from other countries and other labs, and we can verify kinship. There are also some commercial kits for microsatellites. Some of them on the animal side, but most labs design our own systems because of the high costs, and there are not many commercial options regarding microsatellite systems. How can we use DNA profile once we have it at the lab? The main use is this identification power, because I already have a barcode from a genome, from a sample. I know what that the plant is such so and so, that the seed comes from so and so. So identity is already a huge value. That same identity can be used in different processes, such as quality process, traceability verification processes, clone verification. This is very much used for eucalyptus. Also, microsatellites are used in the forensic area for humans, or to solve cattle theft problems or cases. For instance, we received some reports where cattle has been stolen, and we actually compare these samples to other, for instance, areas or shops or housing units, and we compare DNA samples in order to reach a conclusion. Genetic fingerprinting can also be used to analyze populations depending on the markers corresponding to each breed. We also verify the different types of products. Many times we receive seeds that belong from a pool of samples to identify if there's just one clone of variety. We also identify varieties through microsatellites. We identify barley. We have a database with 15 references that are now being used in Uruguay. So when we have the genetic profiling of the sample, of the relevant sample, we know which variety it belongs to. This is an example of the plant I was telling you about. This is kinship verification. The first row shows the mother plant with 4 markers. Of course, it's impossible to see the numbers here. The one in the middle is the product, and number 3 is the alleged father. I think that in allele 12, we can see the match with the mother and father. And this is what we do to verify kinship. We analyze each of the 10, 15, 20 markers to verify this with a 99% accuracy to see if there is kinship or not. Also regarding population analysis, we can group individuals according to similarity, or we can also analyze breeds. What happens at the lab in order to have this DNA profile? Once the sample enters the lab, it is coded with a single numeric barcode that remains with the sample until the report is prepared. DNA is extracted from each sample cells. We receive different types of matrices. In vegetables, most samples come from leaves. We have also received different types of flowers, pollen, grains, seeds. We have quite a large number of types of samples, and depending on the type of sample, we will implement different methodologies through commercial kits or our own kits, and that also impacts on the final price of the study. Then we quantify the quality and we quantify the sample, and we need a sample that is of good quantity and quality. Then there is PCR amplification. PCR is the most famous piece of equipment, as Mariana has already mentioned. And as you can see from the bottom image, each box is a repeat. These are the regions to the sides that are the areas that are important where we design the primers, and through the PCR equipment that changes the temperature, we copy several times the region that we want to clearly see. Once we have the PCR product, we run electrophoresis in an automatic sampler, and it separates the different sections according to size. This is a full profile here of an animal. In gray at the top, and you can see the markers, the alleles, and the numbers as well. That full number is the DNA profile that is unique to that sample being analyzed. We use an AVI 3500 machine with 24 capillaries that can process 96 samples in an hour and a quarter. These are all in-house panels. They are our own panels. They are not commercial kits. They were actually prepared following the guidelines of the International Animal Genetic Association and other switches create based on science. And the idea is to have polymorphic samples or markers, and there we can put together a panels that can distinguish samples or individuals that belong to the same family. That is the key to our panels. The ones that are mostly represented are, for instance, eucalyptus, typification also in cattle. We work with rice, sheep. We have worked with mosquitoes as well. We prepared this panel because a requester asked us to prepare this specifically. We now have around 200,000 DNA profiles in the system at the lab. These labs just are recorded, and we can always go back to them to run new kinship tests. at Genexa, we want to ensure the quality of results. So we need to ensure traceability assurance. And this is what we focus on the most. So we admit the sample, the sample is coded, the coder remains throughout the process, and then we identify the samples and we issue the reports. We have our own sample management system. It has different functions. For instance, it checks kinship relations, it identifies clones, potential fathers or mothers. And this is what the software does. I would like to introduce you to the team. There are not many of us, but we are very good. Andrés Pereira, my partner, Rodolfo Bresno, Ana López, Mariana García, Florencia Arezo, Rodrigo Durán, and Sofía Ramírez, and also Santiago Sousa, Matilde Cortabarria, who are not in the picture because they have just joined our company. So I remain at your disposal in case you have any kind of question. Thank you very much for the invitation. Thank you for participating. Your presentation has been very clear. So for those of us who have science training who are trained in science, it's great to be able to see how these molecular techniques are applied. And now we are conducting this type of analysis, and this shows that there are many people interested in the topic. I believe that INASE also provides the support for field work and for these tools in particular. Thank you very much for your presentation. We have a few questions here. Luisa asks, do you plan to expand the variety identification technique to other species? Some I wasn't even able to show because we have developed them on demand. And we're always open to developing new systems. It depends on the client. Of course, we can discuss this. We keep developing new panels. So that's not a problem. It's just, we will need to talk it over. Do you issue analysis certificates, and do they match the sample that you get? Do you have commercial lots? Do you conduct a sampling so that the analysis corresponds to the lot and not just to the sample? Actually, the DNA profile is unique. When you analyze a mixed sample, those peaks appear mixed up. This tool is valuable because we can determine the unique profile of the sample. Many times we receive sample pools, and we report the existence of one clone or several clones, or if there is a combination thereof. And this is part of the report we prepare. Great, thank you very much. Your replies have been very clear, and you have Rosina's contact information in case you need to talk to her about something.
So like in the former blogs, we will make a question to this members of this panel so that they give their opinions. So people that are from the audience, they can have a global idea on a topic that it's very important. So the question is, what's your opinion on informing the analysis, trading analysis, commercialization for seeds, the identification and the purity? Do you think they will be compulsory in some moment? Alejandra may start. Is it on? Yes. Okay, I will start from the ministry. So from our position, we think it's important, this information. It wouldn't be possible to check 100% of everything, but we must control. It doesn't matter that we can trust what's coming, but we should check, make at least random checkings. And this is very important. And in specific cases, to define according to the markets, we have to protect. Uruguay is importing products, and there are some species that if the market needs one event or another, not a GMO and so on, we have to control to take care of our markets. So we believe it would be good to do that. I don't know what Mariana is going to say about controls, who is going to pay for it, and all the logistics that we have to develop. But as a ministry, we believe it is important. Thank you. Well, thank you very much. Excellent. Rosina with the floor. Well, from my position, it's a little difficult to say because there is a conflict of interest because we are a service lab and we would be very pleased to run all the tests that are necessary. We have the tools and they are available. We are not the ones deciding about it, but I think that gradually this is something that should be done. Any type of control is always adding to the picture. Mariana, what do you think? Well, in fact, I agree with them. I believe this is something that sooner or later it's going to happen because of the quality requirements internationally in the country. We have the technical capacities to develop it. It was very clear in these three presentations. And I think it goes hand in hand with the specialization of the sector. And we probably wouldn't be able to start handling 100% of all the traded lots, but we could start. And I think in a not so far future, it will happen. So we agree with the opinion of something that is not a requirement, but it could be a requirement soon. So thank you very much, the three of you, for your presentation, the preparation, and to be here today. Thank you very much. And we would like you to participate in the last part of our work today. Thank you.